Now we will be focusing on how a scientist will try to understand the sulfur cycling metabolisms and its taxa. So it starts with site description and sampling, followed by sampling locations. Then they physically go to the place and the physical and chemical measurements are taken place. Then sample is collected and some of the in situ measurements, also temperature and other things, are measured. And once the sample is collected, there are two types of samples. One is a sediment sample, another one is a water sample. So what happens to the sediment sample? So once the sediment sample is collected, from that DNA is extracted from the sediments, and after the DNA extraction, it is put to RNA extraction, followed by quality control, then meta-transcriptome sequencing, and finally transcript analysis is carried out. So this is one type of biological analysis. And then after the DNA extraction, it is subjected to metagenome sequencing. Then quality control and processing happens. Then it is actually taken for the assembly and annotation, and then bin classification, then gene identification takes place. So this is another path the scientists carry out. And in the quality control and processing step itself, next is the data integration and comparison takes place. From the data integration and comparison, it is taken to the final results for publication. And the water sample, whatever is collected, in fact, after the data integration and comparison, people also carry out phylogenomic analysis, and from the bin classification also people do the phylogenomic analysis. So these are all the two ways by which the entire species is sequenced. And then water sample is first measured for quality, and then it is filtered water storage. It goes to the filtered water storage for future analysis, and then from there people study about the total carbon and nitrogen measurements are carried out, and the other set of sample is taken for the biological profiling, that is 16S RNA sampling is done. So the first step is similar to the DNA extraction in the sediment also. The DNA extraction for 16S RNA first happens, then PCR amplification takes place, then sequencing libraries are prepared, then sequencing happens, then finally ASV analysis is done. Then after the comparative analysis, the final results are published. So this is how the various steps that are required to characterize the sulfur cycling metabolism and the taxa that is being carried out by a scientist. Here we are looking at the chemolithotropes in the Arctic environment. We will start with what are the relationship they have between different species and what are all the processes they are actually involved in. So all those things we will see. The first one is the symbiotic relationship, that is how two species coexist together. They engages with other microbes, so one microbe engage with another microbe, and also they enhances nutrient availability. So it is like I supply to you, you supply to me, like that. There is a mutual exchange or symbiotic relationship happens between various chemolithotropes. Then temperature and light adaptation is very important. What is this light adaptation? Basically, if you look at the Arctic region, we have polar nights. Polar night has no sunlight. That means more than 3 or 4 months, the complete darkness is there in that polar region, especially in the Arctic region. So that's the reason the light availability is very low. So how do the species survive under these no light conditions? So that is what, and also the temperatures are very low, very cold, freezing cold, even lower than freezing cold, minus 20 degree in some places. So how the chemolithotropes can survive under these extreme conditions? They adapt to seasonal changes, and also some utilize light for photosynthesis. This happens only in the summer. And they also have antifreeze proteins, which we have studied earlier also. So they stabilize the cells during freezing, and they also produce, the species also produces antifreeze proteins. So that is what psychrophilic adaptations when we talk about. Then what is the metabolic flexibility these species have? They are involved in carbon fixation for organic matter. Also they utilize inorganic compounds for energy. So both type of carbons are basically fixed. Then their psychrophilic adaptations include membrane remains fluid, so they adapt to this particular conditions. And some of the enzymes function only at low temperatures. So these are some of the adaptations the chemolithotrophs have. And they also go into dormancy and resilience. That means if the temperature is very low, they just go into the dormant stage. So what they are, how do they do that is basically in their system, they reduce the metabolic process. So the metabolic down regulation during harsh condition happens. That means they slow down the metabolic process. Similarly, they form spores or cysts. So by that they can also survive the extreme conditions. Then they also form biofilms. The biofilm helps them adhere to surfaces. The mollusk and other species, if you look at, they actually stick to the vessels and other thing. So vessels mean basically the ship and boat and other things. So similar to that, these chemolithotroph also produce biofilms for attachment, and they trap nutrients and creates a protective microenvironment. Trapping nutrient means like a cobweb. They can also capture any nutrient which is flowing through near their area. They want to capture it, so they use the biofilm for that particular purpose. And also they want to protect from the microenvironment. For that also they use. So here is a comparison table about chemolithotrophy and photoautotrophy that is happening specifically in the Arctic region. So these two processes requires energy. So how do they get it? For the chemolithotrophy, the oxidation of inorganic compounds is the main source of energy, whereas for the photoautotrophy, light energy for photosynthesis is the main source of energy. What are the chemical reactions that are happening? So in the chemolithotrophy, we have hydrogen sulfide oxidation and ammonia oxidation. Those reactions are given here. In the case of photoautotrophy, it is basically the photosynthesis, though this happens mainly in the plants. Then how do they fix the carbon? The Calvin cycle is the path in both the cases through which the carbon is fixed. How do they adapt to the Arctic conditions? The chemolithotroph does not rely on light. This is required during the polar night. So what they do is they utilize the inorganic substances available in the extreme environment. Whereas the photoautotrophy, this is limited by availability of light during long polar nights. Their efficiency is affected by extreme cold. That means they go into like a dormant stage. Some of the examples are given here. Arctic mosses and lichens adapt to low light conditions, which is the example of photoautotrophy. Whereas in the case of chemolithotrophy, sulfur oxidizing bacteria and ammonia oxidizing bacteria are the examples. What are their roles in the ecosystem? The chemolithotroph drive the nutrient cycle, basically nitrogen, sulfur and iron cycles. They also support the ecosystems in nutrient poor and anoxic conditions. In the case of photoautotrophy, they are the base of food chain in the environment. When sufficiently light is present, they are the primary producers and they produce oxygen. And what are the ecological implications? The chemolithotroph thrives in the cold environments with minimal sunlight, and they also influence the carbon and nitrogen cycling and also affect the climate dynamics. The photoautotrophic species, the growth and productivity are seasonal because they grow only during the summer. In the winter, they don't grow because there is no sunlight. They contribute to carbon sequestration and oxygen production during active growth, because in the photosynthesis, we know carbon dioxide is converted into energy and carbohydrates. So that's the reason they contribute to removal of carbon or carbon sequestration. So how do we study all these things? Here are some of the mechanisms and the pathways by which we can understand. The Arctic climate change mechanisms include temperature change, permafrost thawing, ocean acidification and salinity variation. These are all the four climate stressors we have seen. They are the same thing. So they impact the temperature change impact the microbial metabolic rate and process. So that means if it is very cold, the species reduces the metabolic rates. And the permafrost thawing releases inorganic substances for chemolithotroph. So that is really a good one. Ocean acidification influences the marine chemolithotroph communities. And the salinity changes also affect the microbial metabolism in coastal and marine environment. But Chemolithotrophic specific pathways, when we talk about oxidation of inorganic compounds and electron transport chain and carbon fixation, are the pathways by which they contribute. So how do we study that? We can do the sampling molecular techniques, biochemical assays or microscopy. So how do we do the sampling? We can do soil and sediment sampling. We collect samples at various depth and locations. Ice core sampling can be done. That means extraction of samples from the ice layers containing trapped microbes. We can also do water sampling like collection of fresh water and marine environments. The molecular techniques include DNA sequencing, qPCR, that is quantitative PCR, and metagenomic analysis. And the biochemical assays include enzyme activity assays, stable isotope probing, and microscopic studies include fluorescence microscopy and electron microscopy. So the fluorescence microscopy can be used for the visualization of live microbial cells, whereas the electron microscopy can be used for the examination of microbial structures. So what are the different models? One is the ecological model, and we can also use statistical tool to understand that, and the geospatial tool to locate where they are. So the ecological model include biogeochemical models and microbial community models. In the biogeochemical model, we can simulate the nutrient cycling and microbial process. In the microbial community model, we can predict the interactions and dynamics of chemolithotrophs populations. What are the different statistical tools we can apply? One is the multivariate analysis, another one is the time series analysis. So the time series analysis study helps in the understanding of microbial activity over a period of time. And the geospatial tools include GIS, that is Geographic Information Systems, and remote sensing. Both of them can be used for mapping and the distribution or monitoring of the environmental changes that is happening in the Arctic region.